Welcome to this explainer. Okay, today we're diving into basically the ultimate mystery of the Cambridge AS and A-level biology at 9700 syllabus. You know the scenario. Why do incredibly dedicated students, the ones who literally memorize the entire biology textbook, still end up losing crucial exam marks? I mean, you put in the hours, you know the facts cold, yet that top grade somehow just slips away. Well, today we're going to change that by revealing exactly how examiners actually think. It's honestly a frustrating reality, isn't it? We've all felt the sting of opening up our results and seeing a B when we stayed up all night studying for an A-star. But here's the thing. The answer to this question doesn't just lie in what you know. It's about exactly how you're writing it down on the page. In this explainer, we're going to expose the invisible traps set by examiners and hand you the definitive playbook to bypass them entirely. Because at this level, success is all about your methodology, not just your memory. Take a look at this examiner's blueprint we've got up here, and I want you to specifically focus on the structure of that triglyceride molecule over on the left. This visual brilliantly illustrates a core truth. Examiners aren't just sitting there hunting for isolated facts. No, they are looking for a highly specific procedural sequence. So when a question asks you to describe or explain a biological principle, like, say, ester bonds and lipids, they absolutely expect a systematic approach. See those red warning signs on the right? If you use imprecise vocabulary, or if you just dump a generic textbook answer instead of tailoring it to the specific scenario they gave you, well, that's a guaranteed way to bleed marks. To counter those traps, you need a rock-solid system. Enter the ultimate four-step answer framework. This is how you systematically capture every single available mark. Step 1 is concept identification. You've got to clearly establish the core biological principle first. Step 2, evidence extraction. Literally pull specific values, trends, or labels right out of the exam diagrams or tables. Step 3 is scientific reasoning. This is where you connect your evidence to the concept using causal-because relationships. And finally, step 4, examiner wording. Lock in your answer using the exact terminology and units approved by the official mark scheme. This sequence right here, it moves you flawlessly from a basic concept to an absolutely undeniable A-star answer. Section 1. Precision, beating terminology traps. Now that we have our framework in place, let's look at where students tend to stumble the most. When we're reviewing Section 6.2 on genes and protein synthesis, draw your attention to the top left of this translation diagram. Notice how we're clearly contrasting the mRNA codon with the tRNA anticodon? Well, according to principal examiner reports, mixing these two up, confusing an mRNA triplet with a tRNA triplet, causes a fatal cause and effect error in exam responses. It happens all the time. Lower performing students will actually reverse the relationship, somehow suggesting that the polypeptide chain creates the mRNA sequence rather than the other way around. Defining your terms with absolute clarity, like stating that the tRNA anticodon binds to the complementary mRNA codon via hydrogen bonding at the ribosome, is totally not negotiable. So what it really comes down to is understanding the vast difference between a generic answer and a true top-band answer. A flawed mid-tier response might just vaguely say, tRNA binds to mRNA to make a protein. I mean, sure, it's not completely wrong, but it completely lacks specific terminology and a precise mechanism. The top-band examiner-approved response, it bridges that gap by specifying the exact chemical interaction and the physical location. Doing this proves to the examiner that you haven't just memorized a bullet point summary, you actually understand the underlying biological mechanics. Let's quickly fire off the top 3 red flags pulled straight from principal examiner reports that you absolutely must avoid. Number 1, vague terminology. Close enough vocabulary just won't cut it. Number 2, missing context specificity. Basically, if the question is specifically about a plant cell, do not give a generic answer about all cells. And number 3, ignoring units and anomalies. If you forget to check your measurement units in a data table, or if you just gloss over weird anomalous results during a practical investigation question, you are going to hemorrhage your score. Don't do it. Section 2, mastering the method mark. Moving past terminology, we are entering a really critical phase of exam execution. Simply jumping straight to the final conclusion, yeah, that's rarely enough. To see how this builds, let's investigate this graph on enzyme activity. Check out the curve on the right showing peak activity at exactly 40 degrees Celsius. This beautifully exposes the missing method mark, trap. A rushed student might just write, as temperature goes up, the enzyme works faster, until it stops. Bam, marks lost for being vague and generic. To actually secure the method mark, you have to process the data out loud. You have to extract specific figures, like explicitly quoting that 40 degrees Celsius peak.
and then explain the exact trends using principles. For example, mentioning how increased kinetic energy leads to more frequent successful collisions. The examiner needs to see your thought process.
I cannot stress this rule enough. Always show you're working. Seriously, marks are very often awarded just for having the correct process, even if your final calculation or conclusion is a little bit off. You have to execute your analysis using clear notation and highly structured scientific reasoning. This is exactly how you scoop up those hidden process marks that bump your final grade up into the highest possible tiers. Section 3. Higher Order Evaluative Thinking. Okay, once you've mastered the method marks, you are officially ready for the highest tier of the grading rubric. But what does evaluative thinking actually mean in plain English? Basically, it means moving way beyond simple descriptions. It's about questioning the underlying assumptions made in an experiment, taking a step back to consider alternative interpretations of the data, and crucially justifying your scientific decisions with solid, undeniable evidence. You're analyzing the why and the how reliable, rather than just parroting back the what. Let's look at what evaluative thinking looks like in practice. When you're assessing the conservation roles of things like national parks, zoos, or seed banks, high-scoring answers will always evaluate the quality of the evidence. Check out the examiner's evaluation checklist right here on the slide. You actually need to identify the limitations within the provided biological scenarios and use comparative connectives to bridge both sides of a biological argument. We've even got a great little mnemonic hack for you at the bottom right, ZBNFS. That stands for zoos, botanic gardens, national parks, frozen zoos, and seed banks. Using that ensures you won't forget a single conservation role when you're evaluating a complete ecosystem. Section 4, the cross-topic advantage. We're now arriving at the final insider secret, the one that definitively separates the good students from the absolute top-tier candidates. Let's consider the mitochondrial link reaction, where pyruvate is converted to acetyl-CoA. It's a beautifully complex process, but here is the secret sauce. Candidates who actively reference connections between the link reaction and completely different syllabus areas, like plant respiration or active transport, consistently achieve significantly higher marks. Why? Because examiners heavily prioritize the depth of your analysis. When you show that you understand how the formation of acetyl-CoA in the mitochondrial matrix integrates with the broader energy demands of the entire cell, you prove you have a connected, conceptual grasp of biology. It's like putting together a giant puzzle rather than just reciting a memorized list of chemical steps. To really hammer this point home, let's examine Taq polymerase and its use in PCR. Get this. A shocking 9 out of 10 students make an error here when dealing with multi-step biological pathways. No way, right? But it's true. And why? Because they fall right into the trap of treating concepts in isolation. They fail to demonstrate that connected understanding we just talked about. A student might know perfectly well what Taq polymerase is, but they fail to tailor its role to the highly specific context given in the exam question. Or they completely forget to check the required statistical tests, like t-tests or chi-squared, when analyzing the resulting genetic data. Context is absolute king here, and connecting your topics is exactly how you crown your exam paper. Section 5, your ultimate exam takeaway. All right, we have covered a massive amount of ground today. Let's recap the absolute core message of this explainer. Before you even think about writing a single word about animal or plant cells, you must systematically deconstruct the question. Ask yourself, what is the actual command word here? Are they asking you to merely describe the fluid plasma membrane, or are they asking you to explain the function of the rigid cellulose boundary? Always, always check your units. Are we talking micrometers or nanometers today? And most importantly, vigorously apply that 4-step framework: concept, evidence, reasoning, and examiner wording. Dodge that vague answer trap, and make absolutely sure every single point you make is explicitly tied to the scenario they've given you. So, I'm going to leave you with this final, provocative thought. Are you just blindly memorizing biology facts, or are you truly mastering the examiner's playbook? Because true, undeniable success requires a complete shift in methodology. Take this visual framework, keep those examiner red flags fresh in your mind, and literally apply them immediately to your very next past paper. Shift your perspective, practice the process, and I promise you, you won't just learn biology, you are going to completely master the exam. Thanks for joining me in this explainer, and keep learning effectively.